Welcome everybody to my presentation, and my name is David. So now today we will talk about the importance of DNA and genes. Okay, we will just look through the table of content, and in my presentation, you guys will know about the information of DNA, of genes, and also its importance and the process of transcription as well as the translation. And the last one is its application. Okay. Now we will talk about some information of DNA. This is the story when in the first time in 1869, Johann Frederick found the nucleic acid. Then in 1919, the molecular structure of DNA was defined, and also the 4 base in DNA were also defined, and in the next slide we'll talk about it. And then in 1944, 3 scientists, they Oswald, Avery, McLeod, and McCarthy found that DNA can carry the genetic information. And in the middle of 19th century, Edwin defined the base pairing, and lastly in 1953, just 3 years after Edwin defined the base pairing, another 3 scientists discovered the structure of DNA. Still about the DNA. So this will show you about what DNA stands for. The first letter D stands for the deoxyribose. And this substance in the DNA. Then is the nucleic, and the last one is acid. This is the 3 main substances that to form DNA. This is the image of DNA. DNA has 3 characteristics. The first one is the genetic material, and it can control and perform the activities in a living organism. It has the double helix shape, and it makes up the chromosome, which will then make up the gene. This image carries a lot of information about the DNA. So first one we will I'll show you about the 4 base in the DNA. The first one is the adenine. Second one is thymine. Third one is guanine, and the last one is cytosine. This 4 base is the substance to hold 2 strands of DNA together, and will form a chromosome. A lot of DNA will combine to form a chromosome, and this is the process to form DNA into the proteins. where it from DNA will become the RNA, and then become a protein through the two process, and it's transcription and translation. This is the image of a gene. Gene has two parts in its structure, which are exon and intron. In general, gene is a unit of inheritance formed by many chromosomes, as I have said before. Then, like DNA, it also appears in pairs, and its function is to encode the amino acid sequence of a protein. Then I will show you the function of exon and intron. Exon and intron are two parts in a gene, which exon will have the function of encoding for protein, and the intron have the function is to provide a source of new genes. Okay, this is the process of transcription, and as you can see, from double helix shape in gene, then it becomes a straight line, and it will help to transfer the DNA code into the another type of code or message called mRNA or messenger RNA. This is the three stage in transcription. The first stage is the initiation, which is called the base on each of two DNA strands. The next one is the elongation. Is to provide template for new mRNA molecule. And the last one is the termination. Is when the elongation continue to until the RNA polymerase, which is enzyme, encounters a stop sequence. Then at that point, transcription will stop, and the RNA polymerase release the DNA template. Now you can imagine that if you have a code from a computer where it is coded into the two number is 0 and 1, then the transcription will help it to become a letter or a message from a foreigner, and still you don't know what it means until the translation, where you know the meaning of that passage. And this process has a similar function. And in this process, mRNA will act as an intermediate between DNA and protein because that DNA cannot be decoded directly for the production of amino acid. Okay, next, the fifth slide will be about the inheritance of traits. Okay, this can be this I think it's very easy to understand that when you are a child and you receive both characteristics from your mother and your father. And for example, each of them have a very like dominant um, characteristic, and you can have from both of them, like sporty and talkative, and this can define the so you guys can know about its meaning. You can read a bit. In general, the sexual reproduction will give a rise to the new combination of chromosome pair with variation between parents and their offspring. Like in this image. Okay. This for the dominant and recessive trait, or in Vietnamese it's called tính chất, or the gen trội, or gen chi. Yeah. Okay. So the dominant trait often written in a capital letter, and it and the one allele mask the expression for another allele at the same location. It means that when the two children from one parent will have the exactly same characteristic like each other if they are in the homozygous genotype, and the dominant trait will override the trait of recessive allele in heterozygous parent, and the percentage of dominant trait will override the trait of recessive trait is about 75%, where these three image in this table is shown are shown in 75%, and just 25% left is for the recessive trait. And the two main methods of genetic improvement is crossbreeding and selection. The inheritance of trait can be affected by the environmental factors such as the diet, health, weather, age, time of birth and rearing, age of death, and housing, and season of birth. Last one is application. Genetic genes and DNA are used in three fields. The first field is genetic testing, where it has three functions. The first advantage is to confirm or rule out suspect genetic condition. The second is to help determine the person's chance of developing or passing on a genetic disorder. And the last benefit is to identify change in genes, chromosomes, or proteins. Next one is gene therapy or medicine. the first merit of gene therapy is modify a person to treat or cure disease. Yeah, very well known. The second is to replace a disease-causing gene with a healthy copy of the gene, and this can and the scientist or the doctor can use the micro or the nanobots to replace the disease-causing gene. And the last one is to inactivate a disease-causing gene that is not functioning properly. The last upside of gene and DNA is genetic engineering. It can be used in medicine, where it can make a lot of type of medicine, like the vaccine or the insulin. Then it can be also used to do research. And organisms are genetically engineered to discover the function of certain genes. And the third one is in industry. And in industrial application, it is used to transform microorganisms like bacteria or yeast or some insect mammalian cells with a gene coding for a useful protein. And the last one is in agriculture. In this field, DNA and gene can help create genetically modified crops or genetically modified organisms to raise the percentage of success, and can raise the production in the crop. And thank you for listening.